So we are going to start with the eighth lecture of characterization technique. Till now we have studied one technique in detail, that is the X-ray diffraction technique. We have also looked into the instrumentation part. Obviously that's not a complete course in itself, because in X-ray you need to learn a lot of additional things. The sample analysis and the software that we use, how exactly we are going to find out the crystallite size and so on, can be done by using a number of softwares, some of them which are paid, like the experts, okay, high expert score, or we can basically use simply some free softwares are also available, for example foolproof and so on. So we need to be properly trained in these kind of softwares to actually study this technique of XRD and find out the different properties like the crystallite size and so on. But this cannot be covered in this because we are restricted by time. Definitely in future, apart from the syllabus, I will be also uploading some videos on how exactly to use this software by using some practical examples. Okay, but right now we will proceed with the syllabus and look into a more important characterization technique. Rather, today's age, these microscopy techniques are confirmation techniques to check whether your sample has been prepared or not. So today we are going to talk about microscopy. Okay, and the initially, I think microscopy you already might have heard about it. Okay, whenever you were in your biology lab in class 11 or 12, and you might have heard about something as a compound microscope. Okay, but before that, even during your earlier times, during your school time, you might have been uh, talked about or might have seen something which we call as the magnifying glass. Okay, so a magnifying glass is basically one of a microscope. Okay, it's just a microscope, because what happens is whatever you look under a magnifying glass is going to look enlarged. Okay, uh, and this uses just a single lens. So this is an example of a very simple microscope. What happens in a compound microscope is we are actually using a number of lenses in combination to actually have a enlarged and more focused image. Okay, so when we talk about microscope, this factor of magnification and resolution comes into picture. We have already learned that, but anyhow we are going to just have a look another slide based on that also. Okay, so if we just look into a basic compound microscope, what it has is just an eyepiece. Okay, here is your eyepiece, and through the eyepiece you are going to visualize the specimen which is kept somewhere here. Okay, and before the specimen there is one more lens which is connected, which is called the objective lens. So the magnification that we talk about is basically a magnified image which is a contribution from both the objective lens as well as the eyepiece. Okay, so then we cannot observe the specimen in dark. We definitely need to have some kind of illumination falling on it, some kind of electromagnetic radiation. In the visible range, what we do is we basically use a light source. Okay, so we have it kept here, okay, somewhere at the bottom, but then this light will basically get scattered. So one more important component is the condenser lens, which actually allows the light to be focused and fall on the specimen at that particular location. So we have got the specimen holder or the sample holder where the light is getting focused. Okay, so this is how basically a microscope looks, a compound microscope looks, and these are the basic components of a compound microscope, a very simple setup. Now if you look into the factors here, the two important things that we look into is the resolution and the magnification. So by magnification, I hope you know that if you have an object of this much size under the microscope, you will have an impression that this has actually got enlarged. Okay, and you can actually change the field of view by rotating some screws and look into different parts of that magnified image. Okay, so this is what is magnification is all about. So if you look into the ocular, which is nothing but the eyepiece, the magnification is less, but for the objective the magnification would be high, and the total magnification will be a multiplication of both of them. So basically this is the total magnification that we have. Okay, so when we basically use a magnification device, okay, the limit of magnification lies between 500 to 1000x. Okay, so if you can go 1000x into a system, you're basically going into a very deep atomic scale. Okay, and we can study that. What about resolution? From this example, I think it's simply understood that when we say it is not resolved, that means we cannot differentiate between these two close points. Okay, we cannot really say that there is a gap in between them or not. Okay, but when it's a resolved image, that means if you can go into a magnified image and find out that 2 points are easily separated, okay, can be viewed easily separated, we call that it's a well resolved image. Okay. So there are some theoretical limitations to resolution. Okay, if you consider any kind of optical microscope where light is used as a source, it is a 200 nanometer range. So that means if the difference between these two gap is more than less than 200 nanometer, we are going to see something like this. But if it is 200 nanometer or more, if it's a very good microscope, okay, optical microscope, we can definitely be able to see these 2 points separately. On the other hand, if we don't use light and use electron as a source, this limit of this resolution will be as good as 0.25 nanometer. So you can see that there is a large difference. As a result, in place of light, what we do is we use an electron source, okay, to look into the system. So what we have is the resolution is much better. That means that if these two distances are even 0.25 nanometer, I'd be able to visualize this. Okay, but of course the or the construction part is a bit more complicated. Okay, we need to look into different aspects to use a electron microscope. Okay, so if you look into a design of a microscope, there are effectively two kinds of design which you can see here. Okay, so this design is basically either reflective or transmitted. Okay, so transmitted means that the light will pass through the sample and you are going to observe it from observe the signal when the light has passed through it. Okay, and reflected means whatever signal is getting reflected from the surface of the signal, we are going to study that. Okay, so these are the two kinds of design that we have. Now this design applies to both optical and electron microscope. Okay, please remember our aim is to understand electron microscope. Optical microscope, we are considering it in order to draw an analogy because we already know about it. Okay, and we will see that there is not much difference in design. Okay, the concept remains the same. Okay, so if you consider a reflective design, that means the image, basically the signal from the surface is getting reflected. There are the systems which are basically called the stereoscopic system. Okay, now they basically will have a separate optical pathways for each eyepiece. So if you look into a microscope, there are two eyepiece into it. Okay, it's just like a binocular, if you remember, but both of them will be having separate optical pathways. Okay, basically this improves the result. You can see it much more clearly. Even the resolution gets has an effect. Okay, now these reflective designs are upright. Okay, upright. So some designs, like is an example. Okay, these are upright, and then scanning electron microscope is actually a very good example of this, because if you consider the scanning electron microscope, the signal that you study, that means the reflected electrons that are getting reflected from the surface of the sample. What do you mean by upright? By upright we mean that we have the objective, okay, placed above the sample. So if this is your sample here, the objective is placed above. Okay, or we can say in general the detector is placed above. Okay, so the signal gets reflected and we can see. However, in an transmitted design, what happens is we have got the stereoscopic, basically it can be stereoscopic, it can be upright, and this is the transmitted electron microscope. Okay, so if we look into another design which is basically called the inverted microscope, we have the signal falling on the sample, and the study that is done is on the signal which has actually transmitted the sample. Okay, so that is basically called an inverted microscope. Now both of these designs are actually applicable in both optical and electron microscope. So one is upright, one is inverted. So the difference is, in upright, the signal that is being studied is after the sample, or the signal gets reflected from the sample, and the detector is placed above the specimen. In an inverted microscope, the detector is placed below the specimen. Okay, so please remember these factors. That might be a question for the difference. Okay, so when we look into the light microscopy, basically call it as the optical microscopy. What does it do? It uses a visible light and it uses a series of lenses to magnify the object. So what are these lenses? These lenses are basically made up of glass. In some cases even you can have plastic lenses, okay. But then plastic lenses has got its own limitations, but in high-end microscopes and all which uses light as a source, so they will basically use glass, okay. So what happens is, as light passes through the object, okay, how does the image, how we actually conceive the image, okay, in your eyes if you directly look, because what is the detector in this case? The detector for an optical microscope is your eye, because you're looking into the image directly through your eye, okay. So as the light passes through the object of differing refractive index, so every material will have different refractive index at different points, okay. So light will be absorbed at these points in different amounts, okay. They will be reflected in different amounts, they will be diffracted in different amounts, okay. Because of these combination of all these three processes in different amount, you are going to see or look into a object from a 3D point of view, okay. So the difference in intensity of light detected by the viewer or the imaging device, okay. So if you have got a detector or an imaging device, that can be a camera or a photomultiplier tube, we already know. It will basically provide, what does it provide is, it provides a contrast to the sample, okay. You get a contrasting image because the refractive index are different at different points, and the light becomes partially absorbed, partially reflected, partially diffracted, and so on. So it can actually form a contrasting image and then the surrounding, and then it can be identified and seen clearly against a bright background, okay. So we can use a bright background and look into it by looking into the different processes of absorption, reflection and diffraction, okay. Now if we use or look into the image with respect to a bright field or a bright background, this is basically called bright field spectroscopy, and it is basically also known as transmitted or reflected light. We can have a transmitted signal, we can have a reflected signal. In case of a transmitted signal, it might be a transmission electron microscopy. In terms of, for a reflected signal, it might be reflected or scanning electron microscopy, okay. So if we have some kind of mineral, or you are making some kind of nanomaterial, okay, synthesizing some kind of sample in the lab using some kind of chemistry, okay, you can study that under the microscope, okay, and then get me a mineral or material sample, okay, and then we can also analyze it, okay. What we can see is that if there is too much of agglomeration, we cannot definitely see the size, but if it is like the particles which are there are very well dispersed, we can actually in fact see the sizes also, okay. That is also possible. We have got some kind of software to actually analyze that, okay. So if there is something which is called a bright field spectroscopy, there should also be something which is called the dark field spectroscopy, okay. So what do you mean by dark field spectroscopy? Is basically nothing but which is used for a transparent sample. Now if the sample is more or less transparent in nature, you cannot really have a bright field and expect to see the sample. So what we do is we use a dark field there, and this can be actually created by a hollow cone of light such that no light from the illuminator enters the objective. So if you are illuminating the sample, you are not looking into the sample through the objective where the light falls directly into the objective, okay. What we see is only the diffracted light from the specimen, okay. So if you look into the diffracted light from the specimen, we can actually see the image of the transparent sample, okay. So that is what is called the dark field spectroscopy. So now our aim is to study electron microscopy, and we are going to study some of them, okay, not all of them, okay. So we are going to study some of these samples. Some of them, for example, we are going to study transmission electron microscopy a bit in detail and scanning electron microscopy a bit in detail, because this is more or less important, okay. And if you look into the field emission scanning electron microscopy, this is a specialized form of SEM, and field emission transmission electron microscopy is a special form of TEM, okay. We do have something which is called microprobe electron microscopy and environmental scanning electron microscopy. Now this environmental scanning electron microscopy is mostly used for studying biological samples or the samples which are wet in nature and so on, okay. So, but we are not going to study this. What we are going to study is the other four, okay. So these two we are not going to look into right now, okay, in detail. So when we look into the electron beam, okay, instead of light, okay, we are now in an electron microscopy, we are using an electron beam and not light, okay. And then how do we focus it? Because when we use a light, we can use a lens, okay. But what about the electron beam? Because these are electrons, so basically they are charged, okay, and we can focus them using electromagnetic lenses. So you can have an electromagnetic lenses, that means a lens or basically a setup where you can apply an electric and a magnetic field and control the direction of the electrons, because electrons are charged particle and then they will be deflected by these electromagnetic lenses. Okay. The specimen then has to be specially prepared most of the times, okay, not always. Now if we can actually prepare the specimen for an optical sample, it's mostly the preparation process is very simple. We can take up the sample and put it under the optical microscope, but when we do an electron microscopy, the specimen preparation is kind of different, which we are going to look into it in the later classes. Okay. And then this is actually held inside a vacuum chamber. What happens is, if we consider something like an electron, the chances are that if there is some kind of air or other gas within the system, the chances of ionization becomes an effect which we don't need. Okay. So we don't need to ionize the air or the gas within the chamber where the sample is kept. So a vacuum chamber needs to be used. Okay. But in light microscopy, no such thing is needed, because light doesn't have enough energy to actually create any kind of ionization effect. Now the electrons are collected via a follow multiplier tube. Okay. So the electrons which are getting reflected or transmitted from the sample are collected via a photomultiplier tube, and this photomultiplier tube is connected to an electronic system, and an image is formed on either a monitor or a fluorescent screen. So what happens is, if you are connecting it to the electronics and you are basically connecting it to a computer system, this photomultiplier tube will actually enhance the signal or multiply the signal and give you an image on the monitor. On the other hand, if you just have a fluorescent screen, this is going to strike the fluorescent screen and you are going to get a signal, and finally an image is going to get formed. I've got an image of this fluorescent screen and monitor image in the next slide. Okay. Now using these techniques, a wide variety of biological samples and material samples can be viewed and analyzed under the electron microscope. In fact, at some point you can also talk about the size and dimension of a sample under the electron microscope very clearly. So if you use a light microscopy and view an image, okay, of a shape which is in this nature, might be under a light microscope you look into it something like this. On the other hand, if you obtain the signal on a fluorescent screen, you are going to look into the image and you will be having something like this. That means there would be the distribution of the signals which fall on the fluorescent screen, and we are going to get an image something like this. On the other hand, if you are actually obtaining it on a material, on a monitor, the image is going to be more appealing in nature, and you are going to get an image which is more 3D. Okay, gives you a 3D feel. So if you have just a comparison of the three microscopy technique, that is the light microscopy, transmission electron microscopy, and the electron microscopy, the initial thing or the initial part remains the source. Okay, so basically when we use the light microscopy, light is the source. In electron microscopy, basically look into this. Okay, this side we have got the eyepiece lens. Okay, so we are going in the reverse direction. That means that if we look into this initial image here, we are going from here to here. Okay, look into the image, keep upright, this is here to here, but in that we are going from bottom to the top. Okay, so if you go from bottom to the top, the first thing that we are going to see is what is the source, and that is the light source. In a transmission or a scanning electron microscopy, basically electron is the source. How we are going to produce the electron is something which we will be keeping it for the later lectures. And then we have got a condenser lens. So basically this is a condenser lens which might be a glass. Okay, just as a magnifying glass is used to actually condense a signal or focus a signal. We basically use a condenser lens where you can use and apply an electric or a magnetic field to focus it for the electrons. Okay, then we are actually keeping the specimen here, so you can have, let's say for example, a leaf, okay, which you have used in your biological classes to look into the veins of the leaves and so on. And here we are actually going to keep the specimen somewhere here, but please remember we have to prepare the specimen in a certain way. Okay, but this is not the same as in case of scanning electron microscope, because the position of the specimen basically changes. Okay, so next we are going to go into more details of this. Okay, and then we have got the objective lens, and again in place of objective lens we will basically have, in a case of a transmission electron microscope, you can have a projection lens and a condenser lens only in scanning electron microscope. In transmission electron microscope there can also be an objective lens, okay. But then we have to look into more details. I think this picture is not enough to understand this, okay. This will take some time, okay. So the specimen here in this case for a scanning electron microscope is kept at the bottom, okay. So what happens is the signal will come through different optical or different lenses, okay, and get focused and basically fall on the sample. After it falls on the sample, it gets reflected, the signal, okay, and then it is getting detected by the detector, which is connected to a computer system, and then finally we obtain the image on the computer screen. But when we basically consider a transmission electron microscope, the specimen is kept somewhere at the middle, and then after that what we have is the transmitted signal which is actually obtained on the detector, okay, and then we are actually going to get the image. So in this case, in the transmission electron microscope, the detector that is considered is basically nothing but a fluorescent screen. So the transmitted image we have, then we have got the condenser lens which focuses the image, and then it will basically fall on the fluorescent screen and we are going to get the image. So this is a very rough overview of the difference between the three microscopic technique, and we are going to go into more details when we counter them one by one, the TEM and the SEM in general, okay. Now this is another topic which we need to study, but for this we entirely need a complete lecture. So right now I am just going to tell you a basic idea here. What happens when electron basically interacts with matter? So we have studied the topic of interaction of electromagnetic radiation with matter, and then what happens when electron interacts with matter is a number of processes that comes up. Some of the process which I have already studied is something which is called the characteristic X-ray in XRD technique, where we actually have a high energy X-ray, okay, hitting a inner shell electron, displacing it, and a higher energy electron will actually fill the space, and then we will be having some kind of X-ray emitted, which is the characteristics X-ray, okay. But in this case we don't have a high energy X-ray, we have got an electron beam. It can also do the same thing, it can actually dislodge an electron, and as a result of this which you are going to get some kind of X-rays from there, okay. So when an electron interacts with a specimen, a number of effect takes place. I am going to explain each of these effects later in some other lecture. This is not appropriate for this particular lecture. But right now you need to remember a few points, that when electron interacts with a sample, we have got something which we call as the Auger effect. You might have heard about it, and we get from there something which we call as the Auger electron, and from the Auger electron we can actually study the surface atomic composition, okay. So you use these ideas, and please remember what each of these electrons which are the output after an electron beam is incident on a sample helps us to study, okay. And then we have got the secondary electron. The secondary electron is mostly used for studying the scanning electron microscopy, okay. We also study scanning electron microscopy using the backscattered electron, okay, and this will basically give you an idea about the atomic number and the phase difference. We have talked about Bremsstrahlung. Now on deep inside the system we also have the process of Bremsstrahlung. What happens is the electrons will go and pass through the system, but electron is negatively charged and the nucleus is positively charged, as a result of which the electron speed gets decelerated. Because of this deceleration some kind of X-ray is given out, which is nothing but Bremsstrahlung. On the other hand, we also had got something which we call as the cathodoluminescence, which is the electronic states information, okay. So this cathodoluminescence, I think you have heard about the term luminescence, okay. In luminescence what happens is we have got an output in the form of light, okay. Now what is the reason because of which you have light is something which differentiates different kinds of luminescence process, okay. Here we have got the electrons because which we have got a light output, and that is why it is called a cathodoluminescence. And in cathodoluminescence you can actually study the electronic states information. So if you go into the fine structure and so on, we can actually start with the CL studies. There are also some electrons which simply pass through the sample taking some kind of information, and that will be the morphological information which can be used using the transmitted electron, and that is called the transmission electron microscopy. We can also use the electron scattered electrons which are there, and these are called the diffraction electron technique, which we can actually study the structural analysis again, okay. And we can actually obtain a high resolution image using this scattered electron. There are also some kind of inelastic scattering where the energy is somewhere lost. Okay, some energy is lost. In elastic scattering the energy is not lost, but if the energy is lost within the system, this is the inelastic scattering, and then from there we can actually study the composition and bond states, and this is basically called the EELS study. And then there is something which is called the incoherent elastic scattering, which can also be used, but this is more or less irrelevant because we do not study much with this. Okay. So, we will stop here. In the next lecture I am basically going to start with what are the different kinds of electrons that are emitted in more detail when an electron beam interacts with the sample.